Welcome, everyone, to tonight's X Space. My name is Bryce Nichols. I'm a professor of genetics at Rutgers University. I co-founded and was a former board member of the nonprofit Biosafety Now. I'm a former science policy advisor for biosafety in the Office of the Director at NIH, and also currently host of the Science from the Fringe podcast. And it is my pleasure to be joined tonight by a recent guest on Science from the Fringe, Kevin McKernan. Um, Kevin, how are you doing this evening?
I'm good. Can you guys hear me?
Yes, we can hear you. And I'm going to give you a very long introduction because I feel like I didn't do you justice when I introduced you yeah. And you can tell me, maybe some of these things are correct. Maybe I'm throwing in some things that are actually false and you can correct me.
All right.
So first of all, you hold a BS in biology from Emory University. That's correct. What year did you graduate, right?
I graduated in 95.
Okay, right. And then you jumped right away to working in the Human Genome Project at the Whitehead Institute at MIT.
Actually, I have a confession.
Nope.
I spent about 9 or 10 months working at a pharmaceutical marketing company
Oh,
in
no.
Connecticut. Yes,
What?
I confess.
You worked for Big Pharma?
Well,
What?
yeah, I think so. I think that actually played a role. 10 months there as my first job out of college. I was, whatever, a PowerPoint, Excel junkie there, but it didn't last long. I quickly moved on to the Whitehead after that.
Okay, and then in 2000, this is when you started becoming the quote unquote biotech entrepreneur that you're well known for, right? Now you co-founded Agincourt Biosciences, where you served as its chief scientific officer. That's in 2000. That company then was acquired by Beckman Coulter in 2005, which led you to then launch what's called Agincourt Personal Genomics. This is correct, right? In 2005.
That's right. Yeah, there was so we had a skunk works project at Agincourt that Beckman didn't know how to value, and it was competitive with ABI. And at the time, Beckman was actually in a lawsuit with ABI over capillary electrophoresis and heated lids and thermocyclers. And so they spun that out during the acquisition and sent 19 of us across the hall to a new company called APG, or Agincourt Personal Genomics, and they became 49% shareholders with our board, who became 51% shareholders. And we had an NIH grant at that point to build a sequencer for around, I think, $6 or $7 million that had Gina Costa and Doug Smith as the PIs.
Okay, and this is when you bought your first Ferrari, correct? Is that when?
No,
No.
no,
Oh, sorry. Okay, that maybe I'm mistaken. Sorry,
yeah,
that came
yeah,
late.
yeah. I think I was driving a Prius at the time. Shame on me.
That's true. Okay, so that was 2005, and then so that company was acquired in 2006, right? It's only a year after you lost.
Yes.
Okay.
Yeah. So in a year, that company was able to demonstrate we could sequence E. coli with a solid sequencer. We presented that at AGBT, which is this kind of landmark conference that happens in genomics every year down in Florida. And it made a bit of a shock because it was the data we presented was like 10 or 20 times more throughput than 454, which was out in the marketplace at the moment, taking the world by storm. And so that created a bidding war with ABI and Illumina for APG.
So I guess one of the issues that came up with people that were listening to our the podcast we did is that we did, I think refer to it as maybe like wonk out a little bit, where we got a little bit in the weeds. And I was going to try to make sure that if there were things that were possibly confusing to people, that I would make sure to flag them and go over them. And maybe this is a good place to start, because what you're talking about is a technology maybe called, high throughput. You call it high throughput sequencing? Is that, or what?
I think it kind of acquired the term next gen sequencing. That is basically what replaced Sanger sequencing. The big 300 million or $3 billion Human Genome Project was run on Fred Sanger's technology called Sanger sequencing. Granted, it was greatly improved by Lee Hood and others who made it a 4-color fluorescent system that the ABI had the monopoly in the marketplace for, I think, since as long as I can recall, working on the Human Genome Project. So
But the key thing, though, Kevin, is that Sanger sequencing, which is we're really talking about how scalable, maybe, or efficient the
Yes.
ability to we're talking about sequencing of a DNA molecule, and the ability to sequence. How would you even? Now I'm getting into trouble trying to explain this simply myself. what's the difference between Sanger and then these technologies, these next gen technologies? Because they did revolutionize genomics.
Yeah, so sequencing Sanger, like if you remember Celera Genomics and the whole race for the Human Genome Project, each of the sequencing labs that were really large labs there had probably 100 to 300 of those 500,000, they're probably $400,000 sequencers.
Uh-huh.
Right. So you're talking to like 40 to 50 million investment just in the sequencers. And those things would crank out maybe 96 reads that were 600 bases long every 3 hours or something.
Right.
So there was a limit on their throughput, maybe 100 kilobases a day. But they had longer reads. Once we had a human genome sequence and a reference, we could take advantage of using shorter reads, knowing what the genome was structured like. And that enabled us to do these short read sequencers where you could do billions of reads in parallel in 24 hours.
Right. So yeah.
And that really changed the landscape of that. That's what brought the cost of sequencing a genome down from 300 million to maybe $3,000.
Right. And the money that you then used on your second Ferrari, I believe, which was 2011. So I got to keep going with the Ferrari. Okay. So then, all right, here's where you get into the more controversial part of your career, right? 2011 comes around. And this, so what I have here is that inspired by inquiries from cancer patients about cannabinoids, you founded Medicinal Genomics in 2011. What is this inspired by inquiries from cancer patients about cannabinoids mean?
So, Solid had a unique attribute in that it didn't have the longest reads. Illumina had a speed on the read length, but we had more accurate reads, and that plays a role when you sequence cancer, because oftentimes a cancerous tissue isn't 100% of the tissue. When you biopsy, it might be 10% of the tissue, 20% of the tissue. So when you're looking for variants in cancer, you have to contend with the fact that there's two genomes in there and one might be a minority. So the reads themselves have to be as accurate as possible, so you can trust every read and not try to derive the answer of what the low percentage of the cancer cells might be in that mixed pool. So a lot of cancer labs were installing solids, and those solid sequencers, they were what you would do in that scenario is you would sequence a tumor and sequence a normal genome, like their blood versus maybe a biopsy from a breast tissue or something, and try to find mutations that differ between the two that would elucidate maybe there's a uniquely druggable angle for the tumor. And some of this worked out with like Matthew Meyerson and Dan Haber, both one at MGH and one at Dana-Farber, where they found mutations in the EGFR receptor that were uniquely druggable with ERISA. And if you had that mutation, the drug suddenly worked. And if you didn't have that mutation, it was a drug that wasn't going to get through the FDA. So this drug rescue concept on directing the drugs toward particular mutations sort of birthed I'd say that was probably in the like 2000. Everything of that.
Is this, wait, Kevin, is this kind of like, you're talking more like personalized medicine where you could take,
Yes.
okay.
Yeah, and that's what I loved. That's the direction I wanted the sequencing to go. That was kind of personalized medicine that had a very sort of libertarian ethos to it that resonated with me, and I saw this world, and this is what Francis Collins really like promoted to sell the Human Genome Project, which is one reason I've got fairly frustrated in how the pandemic was managed because it was the direct opposite of that. It was everyone get the same thing at the same time. But it was because of that frustration, I think back in 2011, where I saw we had the capacity to sequence these genomes very cheaply, but we were still picking from drugs that were all poison to try to separate cancer from non-cancer tissues. You know, you do whole genome sequencing to sort out which druggable mutations you have, and then go to cisplatin, right? And at the same time, I had a lot of friends with cancer at the time. They're like, well, what about these cannabinoids? They're not toxic. And there's all this sort of hearsay that they work. And as I dug into the literature, I'm like, no one's really sequenced this genome. Like, we don't know shit about this plant. And this is a real like crime. We need to go and figure out the pharmacy that's involved in this plant. If it's truly non-toxic and anti-neoplastic, that is something that is being hidden because of prohibition. I'm going to go do that.
So so with this in a way, so you're combining this idea, this concept of personalized medicine. And I guess I'm not sure do you still believe in this topic or this concept where if you know the sequence of individuals' genomes, you can tailor therapies for them?
Absolutely. I
Okay.
mean, it's hard, but there are several cases out there where sequencing has made a real impact on healthcare. I mean, I think the first and foremost example is non-invasive prenatal testing. Like we don't do amniocentesis anymore, which had a 1 in 400 death rate.
but you're also talking, well, but back at the time, maybe just for clarification's sake, was the idea that they were sequencing genomes so that they could tell which people would respond better to a particular drug? That is another form, even though they're all getting the same drug. Or maybe is it sort of two sides of
Well,
it?
I think, there's certainly the academic pursuit of that mission.
Yeah.
Whether you can pull that off in the FDA environment, it's a whole nother bag, which is where that's the wall I ran into, is that we had the tools to do it, but the FDA process, to perhaps pull a drug through that had a genetic predisposition on the label,
Uh-huh.
was incredibly painful and challenging to do. And many pharmaceutical companies didn't want to do that because it narrows the pool of the people they can apply the drug to.
And then what about the cannabinoid? Like, why did you run into problems trying to, look at those compounds?
Well, they're legal. So, in order for me to sequence the cannabis genome, I actually had to set up a BV in the Netherlands to go and purify the DNA and smuggle it back and sequence it. And that I couldn't do on U.S. shores. So I had to establish a lab over there to do the extraction. We actually did the extraction in the Dillon Hotel on the Keizergracht in Amsterdam, with a makeshift DNA purification system that worked in a hotel room. So, um,
This you're sounding this sounds quite cool, I think, is I guess the word I'd use. It sounds like you're kind of a badass, Kevin, even though you're
it
doing
was
like,
not cool trying to
uh,
get a centrifuge through the airport. I'll tell you that.
I didn't
And
realize the sort of the clandestine efforts that you went through to start and launch this company. But then eventually then you were able to report the first cannabis genome, right?
yeah, that was in 2011.
Hmm.
And it wasn't the best genome. It was what all Illumina could offer at that time, because at the time we didn't realize this. The genome is like 70 or 80% repeats. It's very AT rich. It's very polymorphic. So the short read sequencers just made a mess of it. We had like the average contig size was like 3,000 bases or something. It was a disaster, but it was the first survey of whatever it was. So we got a handle, at least, that we knew then that, okay, we need to get these long reads involved, and we contacted 454 and did a project with them. That was an expensive project that maybe improved the assemblies like to 6,000 bases, but it really didn't come together until PacBio came to light. Pacific Biosciences are these single molecule sequencers that can sequence like 20 kb, that are equivalent, that have better accuracy than Sanger, and that didn't really emerge until 2018. So we
Okay.
redid the
Sorry.
genome in 2018 to finally like finish it.
But so, and all along this time, like you're a businessman, you're making money, you're uh, what I don't understand in your career arc here is how do you find yourself embroiled in this controversy, this major controversy now with respect to the work you're doing analyzing, the DNA contamination in vaccines. Why
Well,
would you dip your toe into this is my question.
so for a few reasons. One, the way our company found a business model was we realized the cannabis market. All the states were demanding that you had to do microbial testing on cannabis, which is fair. I mean, that happens on all the food too. And this is an inhaled product, so it makes sense. You've got to be a little more scrupulous on things you inhale if they have, aspergillus on them. So we built PCR tests to measure all that stuff. And when I saw the PCR test come out for COVID, I was like, this is a scam. Like, we'd never get away with this, and even in the cannabis market. Like, they don't have controls in this thing. This is going to overcall people, and then shut down the economy, probably shut down our business. Luckily, for some bizarre reason, they left like the liquor stores and the dispensaries open, so we were considered an essential business during the pandemic. But I still felt horrible for every other small business that was getting wrecked by this. And I knew that the PCR work that kicked off the pandemic was very conflicted and sloppy. And then the second reason, you'll see that the tone in the paper we published with PacBio on in 2018, um, we knew that would receive a lot of friction in peer review, so we didn't bother with peer review. We basically did a crypto funded peer review, the non-traditional way. We basically got crypto funded to sequence the genome, and we did it in the summertime, and then put out a crypto bounty for reviewers, $500 up front, 500 in the back end. Put your CVs public, put all your reviews public, so there's no hiding from this. And let's get this review done in 7 days. And that worked phenomenally well. and got the genome out there such that now it is used it's in NCBI, and it's been referenced and cited and used in Nature papers. So it worked. We probably don't get the credit of having a Nature paper, but it's basically a preprint plus some online peer reviews that people can find on our website. But it was a non-traditional peer review because I understood that system does not like certain narratives, and it's very pharma influenced, and they don't want to see the advancement of cannabis. Now, the first time I did the cannabis genome, Nature actually wrote a hit piece on the project relating it to Cheech and Chong, and maybe 7 years later, they had a full page front cover episode in Nature about the wonders of the cannabis genomes that people were generating. So they did an about face, but it gives you a sense of what these journals sometimes do is they reinforce a pharmaceutical narrative and will shun and laugh at work that's outside of that box.
I didn't realize they actually. So why would they mock the publication of a genome? I mean, I assume that there were things that you learned interesting to the biology of the plant, right? What was there to make fun of?
That it was weed.
Just that it was weed. But
yeah, that was the depth of their content was that Cheech and Chong finally got their genome sequenced. Ha ha ha.
I guess, right. I mean, so is there actually any possibility for, well, is one of the ideas to possibly tailor marijuana to particular people? Meaning that you,
Oh
is
yeah,
this,
absolutely. So that's an important background. So prohibition of cannabis has made everyone breed cannabis for high THC, and that's because it's prosecuted on weight. Just like in prohibition of alcohol, everything shifted from beer to whiskey because they prosecuted on weight. So you might as well ship something that's like 40% alcohol instead of 5. Well, that made everyone change their breeding strategies in cannabis to ship all THC plants, and that comes at the cost of not having CBD there or CBG and all the other cannabinoids. So all the cannabis became high potency type 1 cannabis. In its natural state, those two alleles are usually 50-50. It's normally 50% THC, 50% CBD. And so they just figured out the Punnett square and bred things into the direction of being high potency. So they got more bang for the buck. If they got caught with a pound, it would be a pound that was twice as potent. So that led to a cannabinoid bottleneck in the population. There's 100 different cannabinoids the plant can make, and we were predominantly moving around cannabis that was only THC, and that limits the therapeutic potential because it's psychoactive, and not everyone wants to get completely zooted to treat pain. CBD is not psychoactive. Most of the cannabinoids in the plant are not psychoactive. It's only a few of them that are, and only if you heat them. So it greatly limited the therapeutic potential of the plant through prohibition.
and I see. So, but your company is trying to, breed out different versions of the plant to make them
Yes.
more, okay. So this is,
Yeah, well, and we don't do breeding, but we've sequenced now. We've got about 2,300 genomes now public on cannabis.
wait, Kevin, hold on a second. You don't, you claim to not do breeding, but I know that you're doing it in some hotel room in
Yeah,
Iceland,
yeah,
right? Is
right.
that what's going on? Just be
No,
honest. No,
no.
no.
We one thing that is legal to ship around the United States is DNA. And since 2018, you can ship hemp around as well. But most people are interested in sequencing plants that have some capacity to make THC, but also they're interested in all the other cannabinoids. Like, how do we breed back the other cannabinoids that were bred out over the years? And to do that, you have to have a good understanding of the genetics of the mother plants you're working with. If they have certain genes knocked out, you'll never get there. And so you have to know the parents, and you have to be able to set up the crosses in a way that you can select for THCV is a popular cannabinoid now that reduces appetite, which may be a safer alternative to Ozempic. And that's one that only recently have the genetic markers been published by other people. There's a group in Australia and a group at Phylos and a group at Salk who have found different markers that predict THCV. And that's all that is a shorter carbon chain on the molecule, and it's not psychoactive, and it reduces your interest in eating. There's also CBDV that's being looked at in epilepsy. So to understand these pathways, you have to get a full sequence of a pretty complicated biochemical pathway, if you will. And that helps you chart out the mutations involved and how to direct the breeding in a manner that will give you the right outcome.
So again, though, it does sound like you've got like a lot on your plate here with this company, and it's doing really well, and it's actually, fighting against a lot of the controversy that dogged it early on, and you're gaining acceptance. So why then? I mean, I guess I understand why. Like you just kind of enjoy, you know, well, I think I know what it is. Like you kind of enjoy being a bit of a renegade in this space. But it is the case that doesn't your work looking first at the PCR and now more recently at the DNA, how does it actually take away from your efforts working with medicinal genomics? Does it come
It's
at some cost? Is there a
Yeah,
cost?
reputationally, it has certainly hurt us. We've got a lot of people that send us legal letters threatening me and all types of nonsense. But we don't. Oh, my computer almost went to sleep there. But we don't play in that space, so it's not like we're not like losing revenue in the vaccine or mRNA space or the PCR space because we're in a completely different business that doesn't really have a whole lot of like FDA involvement. If we were a business that was in the that like needed to get to the FDA, I wouldn't have been able to do this. Like that would have been suicide for the company.
By the way, there's another thing I forgot to mention. You did say something which I thought was quite ironic given its reputation, that you're saying that you're breeding a marijuana plant that will make you lose weight because you'll eat less. Is that correct?
I'm not particularly breeding it.
Oh,
We're sequencing a lot of plants for
ah,
people that are looking to figure out the genetics for that.
okay.
So we basically are we can accept DNA and sequence. We don't do any breeding. I
I
mean,
keep accusing you of doing the breed. For some
it's
reason,
okay.
I feel like you're breeding stuff,
Yeah, yeah.
and
I know you want my number in the back end.
I have no evidence of this. To be clear, it
Yeah,
just
no,
seems like you should be. Um,
no. It's difficult. Like if you become a plant touching company, all the bank regulations change.
oh, I see. Okay, so
Most people that want to breed leave the country. They go to Canada or Spain or overseas because it's complicated to do it here. You can and there's some acceptance of it in some of the more legalized states like California. But it's all companies in the cannabis space are struggling with banking access. If you're someone who's selling them picks and shovels like we are, it's a different story. We still have banking, but
well, how is your company funded? Like, do you have like investors, and or are you turning a profit on some of the like reagents you sell? And
yeah. For the genomic services we do and the PCR tests that we sell, and we also sell tests to a lot of the growers who are tracking down plant pathogens. They're not these aren't pathogens that are harmful to humans. They distort the yield of the plant.
Mhm.
Hop latent virus is one, fusarium is another, and so those are things that a lot of the growers want to screen for so that they don't have to deal with infections. The challenge in cannabis is you can't grow it outside. At least THC cannabis, you can't grow outside, so you have to grow it inside of like a high security prison, and that invites all types of environmental issues managing moisture and mold. And they can sometimes get monocultures have to end up getting grown, and they are often from a single genetic line. They're oftentimes cloned. So there's all types of issues that arise with pathogens in the plants when you force these things indoors. And so they PCR screen a lot of these things to make sure they don't have any pathogens that are gonna they're gonna devastate the yield.
I see. Okay, so so can you get into plasmid gate? Because I think a lot of people are fascinated with this topic, especially those who feel, that there's been a lot of wrongdoing with respect to vaccines. I guess can you
Yeah.
explain how you got involved in looking at the DNA in the mRNA vaccines? And
So
yeah.
this was not an intentional project of ours. It was something that we put out that paper about how the original Corman-Drosten paper was a disaster of conflicts and a messy PCR assay that had absolutely no controls in it. And probably exaggerated the state of the pandemic, created fear, and maybe induced all the lockdowns, right? And that was probably devastating. As yeah, I mean, I'm
Maybe Kevin,
fire
actually,
here.
do you mind? Can we talk a little bit about that PCR gate real quick? We're calling these things gates. So PCR gate, you're talking about a paper that came out very early on. How early? Actually, was it
Yeah, it was neurosurveillance. It was God, I'm forgetting the dates. It was probably in 2000.
20?
And
Well, 2020. You mean early 2020? You're like
yeah, it's
maybe
2020.
March or April
Yeah,
2020, and they use.
around that time frame, and the paper ended up on the the protocol ended up on the WHO's website before it even got through peer review, to try to hurry this along into the marketplace. The authors were editors in the journal they submitted it to. The journal is a state-sponsored journal called Eurosurveillance, and they did not disclose the fact that they had PCR companies waiting to monopolize on this once the paper got through.
Actually, it's January 23rd. Looks like
Oh yeah,
2020.
sorry,
Yeah.
I should have
No, I just was checking it out. So, yeah.
very very early. And so what was missing in that is internal control. And when you run PCR, um, you need to have an internal control to tell you how much DNA from the host that you got. Right? If you just swab someone's nose and only look for virus, the swabbing of someone's nose can vary 1,000 to 10,000 fold. There's a paper I think from Tawana that talks about that. That variance means when you're swabbing someone's nose and measuring CT values of the virus without measuring how many cells it came from, you can't determine a viral load. You just have a viral number in space. You don't really know if you're looking at dead virus or live virus. So without an internal, we have to have internal controls in all of the PCR tests that we run. If you don't have internal control, you don't know if the negative is really negative. You could have just like missed the person's nose and got nothing, right? And if you got 1,000 cells and have a really low CT score, that's a really hot patient. If you have, 1,000,000 cells and a late CT, that is a negligent a patient that doesn't matter. So without that reference point, you don't have a viral load, and they were calling all these tests viral loads.
So wait, Kevin, can you go through CT again? Like the cycle,
Yeah,
the concept? Yeah.
the cycle threshold is a number at which the PCR goes positive, and with that number, you can back calculate the initial concentration of the DNA or the RNA. And so when it's really late, like past 35, it's usually noise. It's not a hard and fast rule of 35. It very much depends on the assay, but generally speaking, after 35, you have to be very careful to have tight controls to understand what you're looking at between 35 and 50. There are some assays you can run out to 50 and their negative controls will not light up, but most assays, particularly with RT-PCR, you tend to get some signal from water at 40. And we were seeing papers published with this Corman-Drosten protocol that were reporting positives in the water at 35. All right, so that's a sign that the assay has problems. There's also reported problems in the primer sequences not matching the virus. We found the fact that some of their primers dimerized to one another, which meant you couldn't multiplex them, and some people were. That would create other types of false positives. So there's a host of problems with that PCR assay that got rushed through. It got peer reviewed at Eurosurveillance in like 27 hours, and of course they were on the editorial board, so they jammed it through. Got it on the WHO's website before it was through peer review, so that this would become the standard that their companies would monopolize. And one of the companies, Tidmol Bio, did in fact get acquired by Roche after this. So it was a play to capitalize on the scare. And the higher the positivity rate of the test, the more valuable the company became, because that meant they needed more tests because there was contact tracing in place, meaning if you got a positive, you'd have to then scrape in 3 or 4 family members for every positive. So there was a financial motivation against sharpening the tool to make sure you didn't have false positives.
So, right. Okay. So the key thing here is that maybe it was, well, I guess whether or not it was intentional or not, there was a test, this PCR test, and that's okay for people, perhaps maybe half the people, say half the people know PCR, polymerase chain reaction. It's a method by which people can detect even a single molecule of a nucleic acid, a DNA or RNA, and it involves, amplifying amplifying through these repeated cycles. Um, that's what polymerase chain reaction. You have multiple cycles in which at each step you double the amount of the material that's there. So if you start with one molecule, the idea is that after, the first round there's 2, and then the next round there's 4, and so on and so forth. I don't know if I'm helping or hurting the explanation
Yeah,
here,
no, I think
but
that's
but
fair. And I think the important thing for people to know about what are you actually measuring after a CT of 30 is a little bit unknown, because
well, you could be amplifying something that wasn't what you intended to amplify based on, you're talking about these things called primers, and those primers are used to direct the the amplification to a specific target based on its genetic sequence, I
Right.
guess.
And
So,
ideally, you sequence validate what your tests are hitting, to know that at, let's say, a CT of 20, it Sanger confirms. At
right.
CT of 30, it Sanger confirms. At a CT of
But
35, we can't get it to sequence.
But maybe the big thing, forget the trees and go to the forest, is that you have a test to determine the extent to which there is COVID in the population. And that test is fraught with all kinds of errors, and errors which any sort of it doesn't take much to look at this as a molecular biologist and say, oh, this seems a bit off. And you're saying that there was a financial incentive to rush this test out to the public so that they could monetize the test, right? But as a consequence of using a crappy test, what will happen is you're going to get an over-reporting of the extent of COVID in the population, right?
Yes.
And you'll also potentially falsely attribute people's deaths to COVID that weren't actually caused by COVID.
100%. You're nailing a really important point there. And I think that brings back Carrie Mullis's points on this, which is that a PCR test in absence of symptoms and a clinician involved is really, I think, incredulous to rely on to quarantine people. You start taking away human rights on something that doesn't have a physician looking at it is I think goes against all types of human rights issues and medical ethics. Normally, a PCR test in and of itself is not enough to confirm a clinical state. It's enough to give you a piece of information that maybe this is the causative agent in the person's sickness, but you really have to mix that with review that piece of data with all the other clinical evidence you have. And we turned that off. On COVID, we basically said, let's let CVS and car parks do this, and we'll skip the clinicians. And any positive test means you're quarantined and go test your family, without any regard to the CT scores. They should have had, I think, a really strict CT score. I think a really good paper came out of GDA Riolt's lab to measure this. They went and did PCR across, I think it was like 3,700 patients. And then they plated all of those patients. All right. Plating means you try to grow the virus on cells. I think they're using kidney cells or Vera cells for this. And they could not get anything to grow after CT of 32. So that made them tune their test to, we're not calling anyone positive unless it's a CT of 32, because only those people have virus that can actually be culturable. Everyone after that point has remnant dead RNA floating around that's creating false positives. And this is a really dangerous thing to do in a pandemic, because what we know from now, in retrospect, is that you can be positive, you can be infectious with COVID for 9 days during your infection, and then you're PCR positive out to 90 days. So that's like 10% of the PCR positivity window where you're in fact infectious. And if you go and take that test and apply it to the whole population, 90% of the people that are positive are actually recovered and immune. So you actually wreck herd immunity by quarantining people with something like that, because you're taking all the people who are immune and ripping them from the population. And in order for herd immunity to work, you need the people who are immune in the population. They become defenders of the rest of the population, because the virus hits them and it goes nowhere. So it's a very destructive approach if you don't have a very CT aware test that looks for something that discerns live from dead virus.
So I guess this is on a personal note here, Kevin. One thing I guess I'd like to say is one of the reasons I appreciate you talking about this right now is, for me, as someone who very early in the pandemic was really caught up in all of the fear mongering and was in fact probably someone that would have not really thought much about all of these concerns that you're raising. I think it's really important to kind of go back and have a bit of a mea culpa and say, oh, what was I thinking at that time? And kind of really go through a lot of the errors in judgment that were made and sort of tease those apart and figure out exactly how those mistakes, some of which, there obviously were mistakes. Obviously, there also were deliberate um, things done to mislead the public. And if we don't go back and look at them, and people like myself that were banging, having, out there, praising the lockdowns and championing these tests as something that was, giving, correct answers on the COVID numbers and stuff. If we can't admit that maybe we were wrong, then we have no business continuing on, being, considered any sort of like credible scientists, in my opinion. So that's why I really appreciate these conversations, not just, what you're talking about here with the PCR tests and how that was potentially leading to, miscalculations of the extent of, not only COVID in the population, but also the lethality of it, which then led to, you know, public health measures that were designed around those numbers, which were either in some cases deliberately misleading or just derived through mistaken assumptions. But then also, of course, with the vaccine. So on a personal level, I find I'm a bit appalled by some of the thinking that I had back then. Hearing you really step through this logically and clearly, like how obvious a lot of the things that I sort of took for granted as being sort of rock solid were in fact completely built on, perhaps let's call it a house of sand or whatever. But I hope that this is something that if we do have another, catastrophic pandemic again, we won't do this. But I suspect many scientists haven't learned that lesson. I don't know. I guess, and especially given the reaction that you've had to your recent stuff with the DNA and vaccines, I am not confident that the scientific community has learned much without revisiting this stuff. So that's just sort of
Yeah.
soapbox, I guess, personally.
Yeah. I think the thing for the public to look out for is, never let them have a test that doesn't have a clinician involved to quarantine somebody. I think that's important. Two, pay attention to where the CDC can actually sequence some of these things. You'll note during the pandemic, the CDC was refusing to sequence things that had a CT over 25 because they couldn't get enough DNA to sequence. So 1 to 5% of the samples the CDC would sequence. You could send them in there and they'd sequence to try to track the.
But Kevin, why was that? Why did they not want to? So the point is that, like, you're saying that, okay, well, the CDC is making these bad policy decisions, but what is there now like, they control everything, right? So we can't, say that they repeat those mistakes. What really
Yes.
recourse is there?
So the reason they were saying don't send us something with a CT over 25 was that they couldn't sequence it. It was too small of amount of DNA for them to actually get DNA sequence from, which means you don't really know if it is COVID or something else. They were doing that to populate next strain and to build out these phylogenetic trees of where this thing was going, and arguably that data would be fed to pharmaceutical companies to make new strains of vaccines. Yeah, going forward, there's certainly a change in the leadership at the CDC, which is very welcome because hopefully the people that they are putting in there are more attuned to this and don't have the financial conflicts that existed before, such that they're not beholden to conjuring up large pandemics that give them control over society. I don't know. We'll have to see where that goes, but the public should know that those are some of the tells. When you see papers like this rushed out very quickly, and you see they put contact tracing involved, and they're not willing to get really tight on the CT levels, then they're exaggerating a pandemic. And if they can't sequence what's actually amplified, then they have no idea what the hell they're tracking. They're only sequencing the cherries on top that are really high CTs. Well, that's a mirage. You don't really know what's circulating and creating the positivity in the people that are at these late CTs.
So then eventually, though, you stopped on you said that issue got a little too hot for you and you sort of backed off on the PCR. Is that my recollection from our conversation we had on the podcast? You can
Well, it looked like it was falling on deaf ears, and a lot of the PCR companies were in fact adjusting their tests so they weren't as sloppy.
Okay.
And so the problem seemed like they started to put controls in, like the Thermo kit had like 8 different amplicons that they were targeting after the S gene target failure occurred. And they started cleaning those things up, but they still weren't doing anything to select for live or dead at the end of it. They were still picking up people. And then the CDC put in a moratorium that if you've been PCR positive, there's no point in you getting tested again for 90 days.
I see.
And I think that slowed down some of the pandemic, or at least the their declaration of it. So, at that point, I ended up writing a paper with Peter McCullough on the differences between the mRNA sequences in the virus and the vaccine, because they modified them substantially to make these mRNAs.
By the way, Kevin, real quick, I did just invite, I don't know if she's on her phone, but I did invite one of your co-authors, Jessica Rose,
Oh, good.
who's
Yeah.
she just showed up and I invited her to speak. So please, if you'd like to join as a speaker, feel free to do so, Jessica. I didn't realize you were going to show up tonight. But since you're here, I welcome that you're here, because of course you're involved in this work too. So please feel free to unmute yourself if you're interested. Sorry, go ahead, Kevin.
Oh yeah, so that paper pointed out that the codon optimization they did and the addition of this N1-methyl pseudouridine would induce ribosomal frame shifting, and that paper got.
Can you explain what ribosomal frame shifting is? That's something that perhaps people don't know what the heck that means.
Yes, so when you change nucleotides, particularly when you change all of them, like they changed 800 nucleotides in the mRNA. It's about a 4,200 base sequence, and 800 of those were used, and they switched to methyl use. And having built DNA sequencers, I know if you add a single methyl group to a base, the enzymes go crazy. They see that as a completely different entity, and they don't behave the way you expect. We were modifying nucleotides all the time to try and get the solid sequencer to behave, and every single modification you would make the ligases behave differently, the polymerases behave differently. So when I saw they did that many mods, which was 100% replacement of all the U's, I'm like, there's no way the ribosomes are going to read this thing correctly. They're gonna try to read what is a different base and make errors. And in fact, the literature was loaded with papers that showed this, that when you put this in place, the ribosomes would basically get confused and basically skip a base and frame shift. And that would mean that you were reading out of frame and you would get chimeric peptides off of the vaccine. And that was all just in silico modeled. It was just a prediction. We didn't have any biology, wet lab work that we could do to prove it. Two reviewers signed off on it, and then an editor at Hindawi kiboshed it after it got through peer review. And so a few years later, Mulroney published a paper showing it, that they actually measured this with mass spec in patients, that they were getting like 9% of the peptides were frame shifted, creating unknown proteins inside of the vaccinated people. So that was a bit of a foreshadowing. And when that happened, people started sending me vials that I didn't have any interest in really playing with. I didn't know what to do with them. So I put them in the freezer for a rainy day. And we were sequencing a bunch of RNA-seq off of cannabis plants that were infected with hop latent viroid, basically like 3 tissues in triplicate every single week. And it was a massive RNA-seq project. And halfway through that project, we started getting sequencing back that wasn't concentrated on the genes.
Wait, hey, Kevin, real quick. Jessica put her hand up real quick. I don't know if she wanted to add something there. Go, Jessica, you want to say something? Thanks for joining. Did you? Oh, I'm sorry. I didn't know if you wanted to say, okay, comment on something that Kevin had said. My bad. I'm sorry. Kev, how are you doing, Jessica? Thanks for joining us.
Yeah, I'm good. I'm just here by accident, so I thought, hey, this looks interesting.
Well, he's about to talk about your guys' recent stuff, so feel free to chime in as soon as whenever you want. So,
Okie dokie.
Okay, thanks. Go ahead, Ken.
So I got these anonymous vials, threw them in the freezer. They expired, or some of them did, not all of them. But when this RNA sequencing pipeline was broken, and we're getting sequence that was basically genome wide. When you do RNA sequencing, all your sequencing should be concentrated on genes. You get just where the RNA is. When that doesn't happen, you've either failed to capture the mRNA in the prep, or your DNase didn't work. And so to sort that out, our team was like, spike in a control positive, a polyadenylated RNA, and see if it gets through the pipeline. If it doesn't get through the pipeline, it may inform you where things are broken. Either your magnetic beads that capture these mRNAs that are these poly-T beads are busted, or your DNase isn't working, and you got to figure out which one in order to fix the problem. So I was like, all right, I don't want to have to wait a week and order an RNA from Invitrogen. I'll just grab these RNAs I have in the freezer. that are polydenylated. They should be it should be pharmaceutical grade. That's going to be the cleanest thing I could possibly use, right? So I spiked those in, sequenced it, and our team came back saying, "Yep, we can see it in there. You've figured something out." And but there's this mysterious other sequence there that doesn't match the reference. What is this?
Wait, Kevin, apologies for interrupting, but I do want to go back to this frame shifting real quick. I know a friend of mine just told me that I should point something out really important here, which is, and maybe it relates to, when they're talking about the mRNA vaccines, and they're saying we're delivering a spike protein, right? Like a specific protein is going to be made in your body based on the sequence that we're introducing into your cells, right? Now, what you said about the frame shifting implies that instead of making 100% of a particular protein, you're making 90% of the time you make that protein, and 10% of the time you make something else. Is that correct?
A library of something else.
Right. Okay. So I wonder then, in terms of is that built? Does that mean that is there issues with kind of like misrepresenting what's actually being introduced to your body? I mean, I know that the RNA itself is not changed anything, but the actual product of the RNA is not 100% of the protein that they claimed it would be, but instead you get, only 90% of it. Are there issues with like regulatory issues that emerge from that? Or am I...
In that they were never forced to demonstrate what proteins their mRNA produced inside of human cells.
Right. And so,
they just handed over a PowerPoint saying, in theory, this RNA will create this protein according to the codon table, which didn't have methyl U in it. So that was a fiction. And we don't know how much it's going to make. We don't know how long it's going to last. So there's a couple variables here.
Right. Now, the other thing, Kevin, though, is we still don't know, again, and I think one thing I want to point out, and I think it's really important, is that you're drawing attention to a lot of potential things that are problematic, but they may not be problematic until there's investigation to see if there's harm caused or any issues that arise because of these unexpected things you're finding. Is that a fair way to characterize it?
That's fair. We didn't have a lab to go and interrogate these things.
Right.
We were just doing this through bioinformatics.
Well, I'm getting a thumbs down from Dr. Lin on that point. But I'm not. All I mean to say is that I think it's important to be precise here. And my concern is that you're raising a lot of very legitimate concerns that should be investigated, and the responsibility for that investigation is falling on people outside of the pharmaceutical industry to sort of figure out a way to pay for this themselves. Like, you're not funded to do this. You're just doing this because, right? So I think a lot of the hypotheses that emerge from things that you've uncovered deserve to be looked at. And unfortunately, there really isn't a place for there to be funding to do that, because you wouldn't trust the pharmaceutical companies to do this, because they should do it, and they should have an analysis that says, all right, these 10%.
They don't have any economic motivation to do it. If you give them a liability waiver and you mandate things, none of this is going to happen. Right. So I think that we have to continue to put the focus, the public's focus on that if there is not liability applied to these vaccines, what is the motivation for a pharmaceutical company to measure any of this stuff? If they can just simply hand a PowerPoint to the FDA saying, we use these novel LNPs to put in an mRNA, and in theory, it will make the spike protein. And by the way, don't look at our RNA integrity plots that we handed you that showed 50% of the mRNA was in fact degraded or too long. That's just a process of warp speed. Move on. And maybe we'll clean that up later. So they had evidence in their hands that the mRNA that they were injecting was not pure, and that it had these errors, these transcriptional and translational errors were evident in their RNA. The RNA integrity plots it's an electrophoresis assay that measures what's the length of the RNA you're injecting. It should be one length. It should be 4,284 bases. And they were showing chromatographs that had a huge smear on them. Which is now documented in the literature to be known to occur with T7 polymerase, the polymerase they use to make these things, whenever you have these codon optimized mRNAs, they have these loopback template switching that goes on that creates all types of artifacts. And it's not degradation. A lot of people thought, oh, the RNA is degrading and you're giving them degraded RNA. It's more likely template switched RNA because some of those peaks are longer than expected. And the only way that happens is if the template switches back on itself and keeps extending longer than it should. So they had a purity problem out of the gate, and no one wanted to look at it because they had the pandemic fear, shoved down everyone's throat.
Jessica, did you want to say something?
I suppose. I just wanted to make a comment on the frame shifting issue. It's kind of ironic that the people who might have been subjected to this phenomenon for producing these aberrant proteins might have lucked out in a way because it would turn out that they weren't producing the specific peptides that are actually a part of this spike protein to include amyloidogenic peptides. Not to say that, aberrant proteins with unknown function and folding is a good thing, but it's not impossible that, people might be avoiding certain other problems that you can expect when the spike protein is actually translated so-called properly.
Oh, make no mistake, though, the advocates of this were claiming that the frame shifted peptides were a good thing because it broadened your immune response.
Ah.
Yeah. This is the retort we got after the Moroni paper came out, was that, oh no, that's okay. That means that your immune response will be more generalized and it'll be more
robust.
But again, these are things, right? So as just from a scientific perspective, these should all just be looked at and tested. We should not, shy away from, looking at the consequence of having these, aberrant proteins being expressed when the intent was to produce something that was, nearly 100% of one thing, right?
It is astonishing to me that
there's no curiosity here.
Well, yeah, I think a fear is part of that. The pandemic fear created a lack of curiosity, and on top of that, in any other circumstance, this would have been considered a prodrug because you have something that's going into a cell that eventually produces your drug. It's not the drug itself.
Oh, right. So can you just explain what a prodrug is versus a
prodrug are drugs that your body has to metabolize into the active form, and when you have a prodrug, you have to monitor both the drug concentration and the pharmacokinetics and the pharmacodynamics of that drug. You have to measure what it turns into and how much and at what rate, because the metabolism to the liver can be very slow. And in this case, it's not the liver, it's the ribosome. But the ribosomal conversion of these RNAs could differ person to person. Like they never measured what's the variance in expression of this protein that you get person to person. There could be genetic reasons why some people make more or less spike. And so we need to know the variance of the final product, what's your actual drug, when you give people a certain amount of prodrug. That was never done. They got away with just a PowerPoint saying this is what it should make, and let's hope that it's up.
But this is unfortunate, given we're still not even to the DNA contamination yet. And this isn't. No. What I mean is that I didn't even appreciate the extent to which the actual RNA product was. Also, there were issues with that. One thing, going back to this, you said that there were variations in the length of the RNA, and not just the length. You're actually saying that it wasn't the length per se, it was the sequence of the RNA was also influenced because of this sort of template switching or mechanism that would produce chimeras, which would be combinations of sequences that you wouldn't necessarily expect to be derived from a single template, or wouldn't be derived from a single template.
Yeah, there's two things driving that artifact. One is they massively GC enriched the template when they did the codon optimization. So if you look at the codon optimization papers, they intentionally try to make the RNAs hairpin, fold back on themselves, because that makes them last longer. When they're rod-shaped RNAs, your body has a harder time clearing them. They also put in that modified base, which means RNase L doesn't clear them as quickly as either. But that codon optimization is really what is driving the template switching. And there's a paper from Moderna in 2023 where they mutated T7 polymerase to eliminate that template switching. I don't know when they implemented that in their pipeline, if they ever did, but the date on the paper implies that all of the original vaccines did not have that T7 polymerase, the modified one. So these template switch artifacts are what's driving what people have called blot gate. When you look at their blots that they did,
Wait, we have another gate? There's
another gate. Blot gate, you
I
have to
didn't
look into.
blot gate. Oh, okay, fair
Yeah,
enough.
blot gate. Yes, blot gate. They made some Western blots that look like somebody drew them.
Okay, Western blots, for those that aren't familiar with it, this is a means by which one can detect proteins in cells. These are that's a detection method for proteins. Sorry.
Yeah, they're all the wrong size on that blot. They're all higher molecular weight, and they got away with just saying, well, they must be glycosylated. But it's like maybe a $10 assay. You can get an enzyme from New England Biolabs that will clip that glycosylation off and see if that's in fact the case. They never did that. So those higher molecular weight proteins are probably chimeric peptides that are a result of this template switching artifact that goes on. So blockgate's interesting. There's all types of image manipulation, I think, that went on in that people documented on Twitter.
Yeah, using something called RoboJess, no less.
Yes. Yeah. Right. Yeah. Somehow you're implicated.
Wait,
It's not me, I'm a robot.
Yeah.
I see. So blot gate. Okay, I will definitely have to look into that. But so we've got the RNA, we've got frame shifting, we've got distributions of sizes, chimeras, we've got all kinds of things. And now that's it, right? We don't have any other problems. Oh, wait, we've got some DNA in there too. So can you explain what the issue you started to talk about it, then I cut you off, but can you go back to the DNA?
Yeah, so we found this DNA in there, which is the source of this DNA is from the plasmid they use to run the T7 polymerase in vitro transcription reaction on. So in order to make RNA, it has to read it from something. Now, this isn't always true. There are some LNP products out there like Onpattro that use microRNAs. When they're small RNAs, like 21 bases, you can synthesize those on a chemical synthesizer. They're very pure, and you don't have these artifacts. But when you need to make something that's like 4,200 letters long, we don't have a way to synthesize that on a chemical synthesizer. So you have to get an enzyme to synthesize it for you, and that enzyme needs to be fed a piece of DNA that it reads it from. In the trials for Pfizer, they made that PCR, or they made that template that they're going to feed the T7 polymerase. They made it from PCR. That does a couple things that makes it cleaner. It does three things that makes it cleaner. One, when you PCR something, you amplify it so the background gets drowned out a millionfold. All right, so you probably feed the PCR reaction a circular piece of DNA that has a sequence that you want to make a piece of DNA from. It amplifies that 4,200 bases, and then the other bases that are there get drowned out through amplification. You get a millionfold amplification of your target, and then the plasmid is now down at 1 part per million compared to your product. The second thing you do in PCR is you put a particular chemical group on it called a biotin on the end of a primer, that when your DNA is amplified, it now has a tag on it that a protein called streptavidin has a really high affinity for. And we have these beads in our space called streptavidin beads that are really good at grabbing onto biotins and ripping DNA out of solution, so you can clean the DNA away very easily. Third thing it does is that when you PCR DNA, you really only amplify 4 of the bases. And this is going to probably blow people's mind, but there's actually more than 4 bases in DNA. They're methylated bases. There's methyl C and methyl A, and as a result of that, you erase all of the methylation status in the DNA when you do this. That's going to be important for a later part of this conversation, that the methylation status actually is an important signal for your cells to know what is human DNA and what is bacterial DNA. And when it sees bacterial DNA, it goes apeshit. When it sees human DNA, it assumes there's a different process going on, and it deals with it more in a less inflammatory manner. So those 3 things are really key that were involved in the first process that they did the trial on. They ran the trial on like 44,000 people, I believe, and they used PCR to generate that DNA for that entire trial. Then Retzis Levy, who's on the ACIP committee, and Josh Guchkow, who is in Israel, wrote this paper demonstrating that it was documented in the EMA leak, the Emergency Medicines, the European Medicines Agency is like the FDA of Europe, and there was an email leak there that disclosed a lot of the Pfizer documents that showed that they changed the process after the trial to skip the PCR step. So now they're going to rely on just E. coli growing up this plasmid DNA as a template. And what that meant is that the backbone of the plasmid was now present at equimolar concentrations to the spike sequence. So they didn't drown it out through PCR. They didn't have a means to fish hook it out with biotin, and they still had all the methylation patterns on that DNA. That DNA is now in the shots, and it wasn't in the trial. And the work that we published recently and finally got to peer review with Jessica and David.
Wait, Kevin, can I really quick? To me, this is such a critically important point that, and I think it's been completely dismissed, ignored by at least scientific community. It just doesn't want to talk about it. When I bring this up with people, they're like, oh, okay, well, they just want to pretend that this didn't happen. They literally changed the process by which the trials were done with something that was different than what they administered at scale to the public. We should all be very concerned about that, whether you're pro-vaccine or anti-vaccine. The fact of the matter that there was a completely different thing analyzed in a trial than was given to the population at scale. Now, it's entirely possible that this contaminating DNA it has no effect, right? In theory, right? But the process itself, the fact that it was altered and there's been no accountability for that is, to me, just insane. And I actually, going back to the fact of the matter, the presence of the DNA, as you'll point out, there are a lot of things that the public should be concerned about because of its presence. But I think simply the fact that there was a change in the manufacturing and that was sort of swept under the rug as if, okay, you listen, nothing to see here. I think that's ridiculous. I can't put
all guidelines that they have in place right now.
Yeah.
In biological manufacturing, when you use a cell as your manufacturing pipeline, you can't always know. You can't know all the molecules that are present because it's such a complex soup. And so the philosophy in biopharmaceuticals like this is the process is the product, because you can't, if you change it, you can't know all the other molecules that might be there. Right. For instance, if you take these plasmids directly out of a coli, you have to worry about endotoxins, which is this particular molecule that's in the coat of the coli that creates cytokine storms and anaphylaxis. So most injectable products are heavily screened for endotoxin. So if you move from a product that is PCR generated, where those are a millionfold drowned out, to one where that step is skipped, you now have a much higher endotoxin load you have to worry about. And that's just one of the molecules in there that can be inflammatory. When you make that change, there's another molecule that's hard to measure, and maybe you're not measuring it correctly. And there's another 1,000 molecules that could be there. Who knows? 1 million. That type of change in biological manufacturing can introduce something that you can't measure. You don't have an assay to measure it. You don't know that it's there, and it ends up creating harm. So that's why they have this mantra that the process is the product. You cannot change the process in biologics like this because we know it's unmeasurable. There's too many molecules that could be there that your system is not designed to pick up. So the fact that they made that change, you don't have to find DNA or endotoxin or anything because they broke the rules right then and there when they made that change.
Correct. Yeah, and everyone should be outraged by that. And when we talk about it, well, so the problem is, of course, that it's related to vaccines. And so there's a lot of people that simply, and I used to be like this, would want to defend vaccines at all costs. And I don't know why I used to think like that, but I no longer do. And the key thing here is that, as you said, there was a process that was used to develop a product that was then tested in a trial. And then they said, okay, here are the results of that trial. We can now scale this up. And then they went and changed the process. So they created an entirely different product, which they then gave to the population at scale. This is not okay. There's no way you can defend this. It has nothing to do with vaccines whatsoever in a way. Like
independent of any DNA stuff that we have found, they should have been pulled right then and there. Like that's the end. You made a change. You didn't. You know, they did disclose it to some of the regulators, and the regulators asked them for a 250 person trial, which is a sign that the regulators are statistically incompetent. You cannot find a serious adverse event in the trial that small. I mean, unless the adverse event is like greater than 1 in 250, you're not going to find it. In fact, I'd argue even if they had one adverse event in 250 in the change, they would chalk that up to being it's 1, the control is 0, move on. You know, they probably wouldn't have moved unless they saw like 10 or 20 adverse events. And the odd thing about that study is they never released the data, which makes me think they did have 10 or 20 adverse events in the 250 person trial and wanted to bury the data. So so they tried. There was some effort to say you change this. They acknowledged the change happened, and then they gave them a layup to get through the change. And the layup they never delivered the data on. So you know, read what you will into that. The way I read that is they didn't pass the layup, and so they hid the data.
And if this wasn't a vaccine, or labeled as a vaccine or whatnot, I think there would be way more outrage about this. I think there would be universal outrage about this. I think the fact that this is branded a vaccine and many people, again, I understand the desire for people to sort of like protect vaccines in a way. I mean, I understand it because I used to think a lot like that, but now you're confronted with the reality where the process used differed. You can't defend it. It's indefensible. So stop trying to defend it. This has really nothing to do with the vaccines or the arguments around vaccines. This singular issue is a procedural change that should have been caused the manufacturing and distribution of it to be stopped. As you say, it doesn't make sense. At least I don't see how it's defendable or defensible whatsoever, regardless of your position on vaccines. So anyway, I'll get off my soapbox. We'll continue with that.
Okay, so we go about we find this contaminant, and we get attacked online by everybody saying some of your vials are expired. You didn't do it in the proper laboratory with the proper people and all this kind of stuff. And no one's reproduced it. You're an anti-vaxxer. All this stuff. So that's fine. More people start reproducing it. In fact, it happened very quickly on Twitter as opposed to through peer review. And we had several people step up and do this from Sin Lee, who did it. He did it in a CLIA laboratory, actually, with Sanger sequencing. And then Philip Buchholz did it. And then we saw Didier Riol published a version of this as well. And then it slowly went to Bridget Koenig and to Ulrich Kammermer. And next thing we know, there's like 10 papers. Even a set of kids, high school kids, went into an FDA laboratory as a summer project and found it. And that was the White Oak facility at the FDA. And that paper you can see was the poor students. They did a good job as high school students. Did a very good job, but the paper kind of got the typical language of, oh yeah, it's over the limit, but probably doesn't matter. It's fragmented, and these aren't functional, and let's move on, kind of thing. They got the safe and effective blessing, kind of put on top of the paper, but. If you're detail oriented and you understand what they're measuring, you can see that no, they found something that was like hundreds of fold over the limit, and they did it in an FDA laboratory. And even high school kids could find this on a low budget summer intern affair. So it's well established. It's there. I think where a lot of the scientific frustration is falling is that the methods you use to measure this can give you drastically different results. And that should concern everybody because pharma is very good at knowing how to choose the methods that give you the lowest number possible, which has gone on here to convince the regulators that, no, we've gotten data from the pharmaceutical companies that claim you guys are wrong. And you will have, if you witnessed the ACET meeting a couple weeks ago, they specifically asked this of the FDA, which is, have you yourself measured this? And they said, no, we're just taking the numbers from pharma on face value that their numbers are right and yours are wrong. And Tracy, to her credit, said, we realize now we can't do that, and we're going to look into it ourselves. And this is there's a new guard in town, so give us some time. We have to figure out how to do this. But she's very thankful for the people in the field that went out and did this and raised their attention to it. But so that brings us to, is this DNA non-functional? As you've already said, Bryce, like this shouldn't be there to begin with. Now we're debating the fact that something's in there that shouldn't be in there. What does it mean? That's really not our job. But we've stepped up and tried to highlight for them that the residual DNA that's in there is very functional and very different for several core reasons. The main reason is that the FDA has the language of an allowable amount of DNA that can be in a shot. It's 10 nanograms. It used to be 10 picograms, which is 1,000-fold lower a couple decades ago. After the NCIA went in place, which is this National Childhood Injury Act, or Vaccine Injury Act. Sorry, I'm murdering that acronym. But Reagan put in place a liability waiver for vaccines, and when that happened, they started dilating all of the contaminants that were allowed. They all expanded 1,000-fold. At least the DNA did. The DNA went from 10 picograms to 10 nanograms. This is a number that was measured by Sheng Fowler and Keith Pedden at the FDA as to how much naked DNA one could tolerate in mice models and cell models, and it was based on naked DNA being injected via a traditional vaccine. And that naked DNA in traditional vaccines is not plasmid, gene therapy DNA. It is usually a host cell, like a mouse cell line or an egg, whatever they grew the vaccine in. And that DNA is very different. It's very large, low copy number, and it's being injected naked, which means that your enzymes tend to degrade that stuff pretty quickly if it's not protected in an LNP. So 5 to 10 minute half-life if you inject DNA into someone's arm. The moment you wrap it in a transfection vehicle like a lipid nanoparticle, all of that goes away. And they know it all goes away because that's the only way the mRNA works. If you injected naked mRNA, it'd have no therapeutic impact either. But once they wrapped it in a lipid nanoparticle, it delivers that straight to cells, and it's delivering the DNA straight to cells as well. So now you have to throw out the 10 nanogram limit and start over, because this is a different beast. We're delivering DNA into your cells, not outside of your cells. And once DNA gets inside of your cells are not used to that. They don't like, when they see DNA, foreign DNA inside of a cell, they go berserk because they think there's sepsis going on or something else, or viral infection. And all types of pathways trigger that start a cytokine storm and interferon response. So that's one way in which it is functional. Another way in which it's functional is all the other genes that are in that plasmid are now at the same copy number as the spike sequence. And some of those genes are controversial. They're an SV40 promoter, enhancer, polyase signal, and an SV40 origin of replication. There's 4 pieces of the SV40 genome in there. And SV40 is a controversial sequence because the whole virus, the 5,000 letter virus, not the 440 bases we have in this vaccine, but the whole virus was in the polio vaccine and arguably created a cancer scare. Other people have spoken about that. I encourage you to follow Jack Cruz. He's dug into a huge history on. I think he's down here in the chat. But he's done a big deep dive on the Cutter incident and how SV40 contamination in prior vaccines actually triggered a large cancer wave. There's a lot of debate around that. We can go into the debate if you want. I've read deeply into that now, and I think it's truly carcinogenic. It's just hard to see in the epidemiological data due to shedding. But so those sequences were of interest to us because we noticed that the sponsor, Pfizer, when they handed their DNA plasmid map to the EMA, and we presume the FDA and Health Canada, that they erased those sequences from the plasmid map to hide them. And that's not something that occurs accidentally. It's something someone physically had to go and do to defraud the regulators, because the annotation tools that annotate sequence don't miss this. They annotate everything. So someone went in and erased it and handed them something to hide that SV40 was in there. So anyway, that got us down a deep dive in SV40. I should probably stop there because I think I may have lost the audience.
Well, the problem that I have, well, not the problem I have, but there's so many details, and a lot of this requires, some molecular biology background. But these are critically important things that really should raise a lot of alarm amongst people that are familiar with you. These aren't things that most scientists would have a hard time understanding would be problems, and yet, by and large, this stuff is just being not only just like ignored, but also it's being attacked. Like because this gets back, I guess one thing is a lot of what you're saying, there are there are measurements that you've made and you and others have made that show the presence of these molecules, and now you recent work is showing that you actually have the methylated form of the DNA in some of these batches or in some of the vaccines that have been administered. These are facts, right? Like these are not something that could be in dispute. What then gets in dispute is what is the consequence to people that have taken these. What effect did that have, if any, right? And that's where further experimentation or analysis of individuals that have or haven't had these different vaccines is really desperately needed. But there's barriers to getting that stuff looked at, right?
There is, and I'm not going to go out on a limb here and say that the DNA is causing all the adverse events. I don't think that's the case. There's so many other things going on in these vaccines, but what?
Right. Well, that's right. So this is important, and this is where I think. Well, I think I said to you before in our other conversation. I think you get in the critique people, or that you get in trouble because the implication of what you're saying is that there are all of these adverse effects that are caused by what you've shown, right? But we don't know. I mean, the problem is it's possible that there have been adverse effects because of this, and everyone should be concerned about that. But instead, we're having arguments about whether, like, I don't even know what we're arguing about, frankly. Like, you've raised some very important points, and everybody that even supports vaccines, whatever your position in vaccine is, you should try, be interested and curious as to whether these have any effects when they're put into people. I don't, yeah.
What I've seen is they've tried to create a bit of a straw man argument on this, in that they'll say, well, so what if it gets into the cell? It doesn't matter, and it'll never get into the nucleus. Or if it does get into the nucleus, it will never integrate.
That's not scientific, Kevin. That's not scientific. I don't give it, like, who cares? The coulda, shoulda, woulda. As a scientist, we can do these experiments. If there's a will, there's a way. They're doable.
Very doable.
Yeah.
But they've tried to point the whole conversation to this limited hangout of, prove that it integrates. And if once you do that, then we'll pay attention. And there's a couple issues with that. Is that, one, they're asking you to prove something that is extraordinarily hard to do because you have to sequence someone's entire genome to figure that out. And once you are forced to do that, you now need to get a CLIA laboratory. You're going to need to get an IRB. You need to get some genetic counselors involved because you're talking about sequencing someone's entire genome, and you're going to unveil their BRCA mutations, their ApoE mutations, and all these incidental findings that you now need to consult the patient with. That, sorry, we sequenced your genome looking for integration, but we found all these other things that we're now obligated to disclose. So it creates a rat's nest of red tape. So they're doing that on purpose. The real crime is committed the moment the DNA gets into the cell. There's a whole other pathway that they're not talking about, which is that you have a pathway that when it sees foreign DNA, it triggers a stimulation of interferon genes, which is the cGAS-STING pathway. STING stands for stimulation of interferon genes. All right. And if you do that repeatedly, you will create cancer. This is documented in the literature. Quanidol has this. A lot of other papers show this. If you do it once, it actually may fight cancer, but if you do it repeatedly, it will sensitize your cells to not fight cancer, and you end up getting oncogenic process underway. So the moment the DNA gets into the cells, you're triggering cGAS-STING and sometimes 12 receptor 9 to create mayhem in the cells. And we are repeatedly injecting people with these things for reasons that I still don't understand. Right? If the vaccine worked, you don't need 10 of them. You don't need 2 of them. You need 1. It doesn't work, right? So they've come up with this booster program to boost revenues. That's all it is. It's not a booster, an immune booster. It's a revenue booster. And so that has led them down the path of letting this DNA remain and taking a risk of it being an oncogenic procedure. The other issue with the DNA that's in there, and this is something that I think the future FDA has to pay attention to, is that we can no longer tolerate a 10 nanogram limit of any DNA. That is a ridiculous loophole, because if I put in DNA in there that's an oncogene, at a very low copy number that amplifies once it gets in, your limit is meaningless, right? We have the capacity to sequence entire human genomes. We certainly have the capacity to sequence the lots of the shots you want to give to 5 billion people, all right, to know every single piece of DNA that's in there. And you can start categorizing it as, okay, is this harmful DNA or oncogenic DNA or harmless DNA? Right now, they're just assuming 10 nanograms of any DNA is fine. I bet they wouldn't say that if it were, an Ebola virus or name your dangerous microbe that could easily slip in there that is replication competent inside of a million cells, and that would be a laughable number. So the SV40 DNA, I say this because we know it's a nuclear targeting sequence. We know it's used in gene therapy. It drives DNA right to the nucleus. We also know it binds P53. We also know that this thing is a somatic hypermutability element, which triggers the ApoBec pathway to create mutations in your DNA. So there's functionality inside this small piece of DNA, and 10 nanograms of a small piece of DNA is like 500 billion copies of it. Right? It's not a molarity limit. It's a weight limit. So that alone shows that they're incompetent about copy number of DNA based on nanograms versus, like, they don't know Avogadro's number, right? This is insane. Now, if you read Sheng Fowler's work and Keith Pettin's work on this at the FDA, they were aware of this molarity issue, that 10 nanograms of genomic DNA is, so many thousands of copies, not a big deal. 10 nanograms of a virus is billions of copies, and that's a big problem. And you need to put the limits down at the femtogram levels if you're dealing with a small copy plasmid like this. We're nowhere near femtograms on this, right? So once LNPs are involved, that whole nanogram limit needs to be reinvestigated very critically, because we're now dealing with DNA that traffics right to the nucleus, and it can make more copies of itself once it gets in.
You did say something earlier on in the conversation that I think is critically important, and you reiterated it just a moment ago, talking about, I think, Tracy Beth Hoag's comments at the recent meeting the FDA had. Is there hope on the horizon for, evaluating this as a potential, problem, and changing regulatory, rules or something, or what do you potentially see happening at the FDA to both deal with the past mistakes that have been made, but also prevent this from happening in the future?
I don't want to speak for her. All I know is that she expressed an interest in our work before she got into the FDA, which makes me think she's quite sensitive to it. But I think a broader question is, these companies have had liability waivers for decades, and we shouldn't stop here. We should go sequence every vaccine they're giving. Who knows what's in there? Right, all of them. Like other people, we're not the first people to find a contaminant like this in DNA. This Sing Lee found this. Another vaccine, I think it was the HPV vaccine, he found DNA contaminations. The plasmids were in there too.
So the argument, Kevin, with this is about the incentives the vaccine manufacturers need to have to make vaccines, right? Do you have a position on that? Like, I understand, and I think most people that are presented with this issue do agree that, that waiver presents a problem, right? But
yes.
But the counter to that is, of course, that, well, the vaccine manufacturers we won't make anything for you now if we don't have the waiver. It's a similar. Well, it's a similar.
Like, you can't make a product that doesn't, like, the market should sort this out. If you can't make a product that you don't get sued into oblivion for, you shouldn't make the product.
That seems like a good argument. That seems like a good counter. Like you would. Yeah, it's kind of hard to beat that logic, I guess. But why is that such an effective argument, it seems? Or at least that's the one that
It's an effective one because they've done a very good job brainwashing everyone to believe that these vaccines are so necessary and you should fear your neighbor. Like, oh, if we don't have a vaccine.
When you say these, you're talking, well, so the other issue, of course.
In general, all vaccines are immortal. They are the emblem of scientific achievement. And
I think it was Paul Thacker that I think who wrote an article called, like, "Vaccines Are Magic" or something like that, which speaks to the way in which that the word vaccine has sort of transcended its meaning as, like, like everything's put into one box.
These things, which are arguably gene therapies, everyone wants to reclassify themselves as a vaccine now so they can have liability-free access to the population. It creates.
Is there talk of marijuana as a vaccine? Have you thought about this?
That's a good idea, actually. We should talk off.
By the way, yes. Because I
What's
think
funny
you're
about all
onto
this is
something.
that they used cannabis to entice people to get shots. They had a Joints for Jab program in Portland.
Please tell me you're
No,
making that up.
look it up.
Oh,
Google Joints for
oh
Jab.
boy. That's okay. That's pretty depressing. Jessica, did you want to say anything on this?
Actually, I think it was probably pretty uplifting, but that's okay.
Oh, right. I didn't know. Jessica, I was going to wrap this up in a little bit. I don't know if you wanted to say anything before I sort of close this space.
No, I've been enjoying listening. Basically, on the liability immunity thing, I mean, if we can get one of these manufacturers on fraud, which I think, you know, it would be hard to do because you have to prove willful misconduct and there, you have to prove intent. But quite frankly, I think with all the documentation and all the admissions and all the evidence that is already written down, we can do this. Um, they have, I believe that at least Pfizer has committed fraud here, misrepresenting a product, claiming it's safe and effective for so long, still doing so when clearly, there are harms associated and or caused by these products in not just a few people, in a lot of people. So I think if we could get the ball rolling, even as a prototype situation, um, have liability immunity removed, and all the things that might come with that, which, would include civil liability exposure and class actions and criminal exposure, et cetera, et cetera. I mean, it could really change the milieu of this vaccine monster that's taken over the world. I mean, it's more than an industry now; it's a monster. It's like people people don't even flinch at the thought of getting a shot every year for something that you can't make a shot against that's going to help you. It's not funny, but I mean, Anyway, that's all I wanted to say.
On that point, Jessica, I want to credit a lot of the people who dug up that information. That's probably going to be very critical
Yeah,
for the fraud case.
over here.
Yeah, Scoops Magoo is here. Follow him. He's got a lot of work. He ATIP'd a lot of the stuff out of Health Canada, which demonstrates that they told the public something different than what they told Pfizer. So they went public saying these fragments are too small to matter and they're not functional. And Scoops found their emails and showed that no, in fact, they told Pfizer to get rid of this and that they don't know the length of these things. So we know the length because we've done some Oxford nanopore sequencing on these things. Some of them are 3,000 to 4,000 bases long. I think Phillips found one that's 5,000 bases long. So they're big pieces of DNA in there. The other people are, I think Tamara Ugolini's done some work on this. Sonia Elijah's done work on this. Marianne Damassi's done work on this. Rebecca Barnett's done work on this. So there's a lot of really good reporters out there that have been digging in with FOIAs and ATIPs to collect information that is going to be really material in that case. So hopeful that if that happens, then the PrEP Act would get dissolved and there would be a liability angle for all the vaccine injured to finally get some of their medical bills dealt with. But to future-proof this a little bit before we end, there is a game that's going on in the vaccine world that people need to know about, which is that they're getting pushback on the adjuvants. So they're removing some of the adjuvants like aluminum, but they're replacing them with things that aren't considered adjuvants today, but should be. You'll see in the HBV vaccine, some of them out there are putting in unmethylated CpG DNA to trigger the immune system. All right. And these aren't in LNP, so they have to use like 3 milligrams of material to get this response. But when you're in an LNP, you arguably need 1,000-fold less. And that's what's going on with these shots is that we have DNA in there that's methylated. And when it's methylated, it's more immune stimulatory. The cGAS-STING pathway in particular does not like seeing methyl A. Methyl A is a sign of bacterial DNA in a cell, and that triggers a huge immune response. and leads to that whole cancer pathway. They also don't like seeing methyl C that's not in the CpG. That triggers toll receptor 9. So these plasmids can be grown in cell lines that don't methylate the DNA, but Pfizer's using cell lines that does. And I think they know why. And they're doing that intentionally because they don't have an adjuvant in these vaccines, and they're relying on the DNA to be that adjuvant. So they probably don't want to clean it up and are going to defend that it doesn't matter till the cows come home because they need it. So I think this is an important avenue for us to focus on is that so the methylated nature of this DNA and the quantity of it being an LNP is they need to investigate what is that doing. What you don't want to switch from aluminum to something that causes cancer, right? That's just I think a bad trade-off, and we'll be back in the same place in 10 years when they swipe all the aluminum out and replace it with plasmid DNA.
Any chance that the vaccine manufacturers will begin to sort of start working with you on these problems? No.
Should we do a polymarket on that?
Are you one thing we didn't get into, of course, is sort of all the sort of, efforts to discredit you and to, deplatform and take your work out, that kind of thing. Have you been served with any legal notices by any of these vaccine?
No, the companies themselves. I think they tend to. I mean, I don't have proof of this, but there's enough other entities out there that were funded by the Biden administration to attack people like us, that they can keep their hands clean of that and let other people do the dirty work.
So I'm not saying anything that's not true. So
yes, yeah. So that's true. That if we said anything that wasn't true, they'd have a case, but they haven't. So they haven't done that yet. But there's no shortage of attack from Pub Smear and from Shots Heard and 77th and the whole Mutton Brigade out there, which has questionable funding and could have come through some type of who knows, USAID or something. But we're fortunate in some ways, or at least I should say I am. I can't say this for Jess or David. You guys have certainly had your share of abuse as well. But typically Shots Heard would go and attack physicians and get their licenses revoked. And so you'd see people like Byron Bridle and Mary Talley Bowden and Kirk Moore and these other people all get throttled by these people to have. There's another one, David Cartland, I think in the UK. They've all gotten attacked on the license front. I don't have a medical license they can take, so they're probably going to try and hit me in other ways. But I had my home address sold on some Twitter space. I've had legal letters sent from trolls that we've more or less diffused and ignored. I've had a lot of that stuff goes on, but I think a lot of it is the paper tiger at the moment. It's just meant to scare a lot of people off the topic, and it's out there. It's not something I can change. There's enough other people measuring this now, and I think that's the power of Twitter and tools like this: is that you decentralize the information, get it replicated, so that there's an army of people that they have to deal with, and they can't go one by one and just Wakefield you individually. They have to now think about Wakefielding a whole community.
Wow,
and that's
now. I love that.
Yeah.
You just dropped Wakefield on the end of our Twitter space. My gosh, Kevin, look at what you're doing.
He's a verb now. He's
a verb. Man. Gosh. Well, with that. Okay. But I actually, thank you so much, by the way, for doing this on the heels of doing that podcast with me. I really appreciate it. And Jessica, thanks a lot for hopping on. I apologize I didn't invite you. I guess and David, I did invite David to speak. I saw he was here, another of the authors on that paper of yours that recently came out, and I congratulate you on finally, getting a peer-reviewed publication on this topic, which is.
And Maria Gucci's here too. She was actually
yes, her and David Wiseman helped as well.
Okay, and so I appreciate everybody's efforts on this. And like I said, I guess part of this for me is just trying to honestly, really just, you can take the idea, like we're just talking about some scientific things here in a way. Like if we focus just on the science, maybe we could get close to 100% agreement that these things should be looked at. It just so happens that this is about a product that was administered to billions of people across the world. So it raises a lot of issues, I guess, with getting some clarity on the, the potential harm that has been caused by this some of the mistakes that were made back early on in the pandemic, and which continued to be made to this day, I guess, right? That's not, we haven't really learned much here. If I'm reading, if what you're telling me is, you had told me that they're still manufacturing the mRNA vaccines in a way that creates problems. Is that?
Yes. Yeah. So we've David's done some of this work. He's measured XBB and JN1 lots, and those are still over the limit. They haven't changed. So and I don't think they will until the liability is pulled.
Why would they change? I mean, if you don't have any, if there's, and that's, right. If there's no incentive, there's no accountability. If you can use all your money to, honestly, I don't even know why people make these decisions. I am just frustrated that the entire scientific community can't just rally and just say, okay, yeah, this is an issue. We should just try to deal with this on a scientific level and forget our preconceived notions about vaccines and whatnot. So that, I guess, is my, I keep coming back to that because when I discuss this with my colleagues, that's the point that I can get agreement on. As soon as it gets related about vaccines and whether you are pro or anti-vax, it just falls off the rails. So that's kind of where
I would end. That's a really good point in that there's a lot of excitement and funding in the mRNA space, right? So you can either be like, oh, we have to shut these people up because they're damaging the reputation of that field, or you can recognize that the damage has been done. The adoption of these vaccines now is so low; it's like 10%. All of the other things that you're building with mRNA, which might show some promise in the future, are going to be held in light of what happened here. So if you don't get on the side of cleaning this up, all of your hopes of using this for cancer are gone. All of your hopes for using this for rare diseases. Like there might be a shot where this type of technology works in a rare disease where you like got a 20-year lifespan and this is your only shot, right? No pun intended. But that's a very different risk reward issue than, all right, we're going to apply this to a virus that the virus isn't benign. I'm not going to go and say it's not there and it's benign, but there's perhaps an argument to be said that the countermeasure was more damaging than the actual risk of the virus. And so that was a bad risk reward. But when you get into cancer and you get into some of these other rare genetic diseases where you need to do some type of enzyme complementation, and if you don't do this, you have a 20-year lifespan, like the Duncan paper had showed. There's a good paper in NEGM where they used a lentiviral transformation of cells that were taken out of a patient's bone marrow, transformed, and they replaced a gene that made these people survive by putting that bone marrow back in. But it came at the expense of 1,000 integrations per genome and cancer. But they're willing to take that risk of lymphoma because they had a lifespan of 20 years without it, right? So there's a place where these things may actually help people, but now they're stained. And unless they get involved in cleaning up the mess, all of that other opportunity is going to be ill-received, I think. So I hope we convince the people who are working on mRNAs to not fight this, but to clean it.
I doubt that's the case. Sorry.
Yeah.
We can end with like a false sense of hope. My impression is that they're going to dig their heels in and only change when they're forced to change, like most people do. It's just sort of human nature.
Yeah.
Hard to admit mistakes, but we can keep trying, I guess. So anyway, Kevin, thanks a lot. Jessica, thanks a lot as well. And I guess with that, I'm going to wrap up tonight's Twitter space. Thanks everybody for joining me. I don't know when I'll be back, but I'll be doing... Wait, Lynn just gave me a thumbs down and what happened there? I don't know why I'm getting a thumbs down from Lynn. But I hope it wasn't because of something I just said. Regardless, thanks everybody for joining. I will be back with another space in the next couple weeks or so. Until then, everyone have a great weekend, a great upcoming weeks. By the way, I have to figure out a way to end these like in a very succinct manner. Kevin, can you give me a recommendation on how to end these Twitter spaces? How should I end it? Because I
can just go to bed. That would end it pretty quickly.
Okay. All right. Everybody, take care. Good night. Thanks. Oh, you are. All right. Well, have a great night, everybody. Thanks for joining.
All right, everyone. Thanks.